So, without further ado, please join me in welcoming Dr. Chris Gibson, the co-founder and CEO of Recursion, a clinical stage biotechnology company harnessing technology to map biology and chemistry and industrialize drug discovery. Dr. Gibson is a graduate of Rice University with degrees in bioengineering and management. He developed the technology and approach that seeded Recursion as part of his MD PhD program, working in the laboratory of his co-founder, Dr. Dean Lee, who's currently president of Merck Research Labs. He did this while at the University of Utah, and after completing a PhD that merged engineering and medicine, Chris took a sabbatical from medical school to build Recursion into the rapidly growing company that it is today. His interests focus on the intersection of technology and biotech and the importance of adaptability and diverse partnerships in driving innovation. So as I prepared my remarks, I listened to an interview that Chris gave for the Biotech 2050 podcast. We're very fortunate to have him here today because I'm sure that he'll share with us some of the material on that podcast. But if you have an opportunity, it's really great listening. So if you don't get enough of Chris, please view that podcast. When asked what contributed to his success, he stressed the importance of surrounding yourself with supportive mentors and incredible people. I believe that we'll hear more about this in his presentation this morning, and I hope that you, as attendees, find those types of people here at the SOT annual meeting. Chris also serves on the board of directors of BioHive, which is a public-private partnership driving expansion of Utah's life science ecosystem, the fastest growing in the nation. Chris is active as an advisor and mentor, both formally and informally, of many biotech founders. We look forward to hearing Chris's insights on this exciting and rapidly changing field and how tech bio is innovating the development of therapeutic. Please welcome Chris Gibson. Thank you. So you're all set with the slides?
Thank you so much. Appreciate it. Perfect. Good morning, everybody. I am so excited to welcome you to Salt Lake City. Our headquarters is just down the street, and it's phenomenal to be able to welcome you to our home. I also was sharing with the group earlier that I love the 8 a.m. start time. I know maybe not everybody does, but for me, 8 a.m. on the dot, this is fantastic. I'm going to try and bring some energy and help you all get excited for the amazing few days that you have ahead. And I want to say a huge thank you to the Society of Toxicology for the invitation today. I'm really excited to share our story. Before I do, can I just get a show of hands if you are currently using ML, AI, or other sophisticated computational techniques in your work? Show of hands, don't be bashful. Amazing. Wow, that looks like half of the audience. Okay, that's great. So let's dive in. I was asked to share a little bit about the story of Recursion, but I should share with you that there are hundreds of companies, both large and small, who are building tools like the ones that I will talk about today. And I think over the next decade, we're going to see an incredible advancement of all of the industries which you all touch, thanks to these tools. So because we run a public company now, I get to have this really cool slide. And if you can all just read it real quick. Perfect. So I'm going to talk now about predicting outcomes from complex systems. And I'm going to start with something you probably haven't thought too much about, and that's the world of technology. And it turns out there's a really simple formula in the world of technology for understanding complex systems. Things like human preference for movies, or what kinds of products you might buy on a website. And that is that you measure with high dimensional readouts lots of information about that system. You aggregate that data, and then you train algorithms to make predictions about the relationships in the data. And initially, those algorithms will be terrible. But the most important piece of this is that you can create this flywheel, this virtuous cycle of learning and iteration by taking those algorithms and making a prediction about how that system might actually work and then testing it. And so let me give you a couple of examples, and we'll start with one of my favorites, Netflix. So back in the 2000s, Netflix was collecting data on tens of millions of people, watching what shows we watched, what order we watched them, what time of day we watched them, how long we watched them, when we turned them off. They were aggregating this rich data set and then making predictions about what shows they should recommend to us. And if you followed along, they've gotten better and better. Through the late 2000s, the early 2010s, they were making better and better recommendations. At least that's how I felt. And what's most amazing is today, Netflix now generates its own content, by and large, based on the algorithm suggesting to them what kind of shows each of us are going to want to watch. So they went from aggregating content to creating content, using this algorithmic approach, this virtuous cycle of learning and iteration. And I think there are some interesting parallels that we'll find when we look at the world of biotech. Google Maps, another great one. Who's seen one of these cars driving down the street? Taking pictures, aggregating information about our world. They look at many different layers of data, actual maps. They have airplanes flying over cities. They've got data from our phones measuring how long we're stuck in traffic. And all of this information is going into the algorithm to make predictions about how we should interact with the physical world. And another one, Amazon, making predictions using all of the data they aggregate from us about what kinds of products we might buy. And in fact, now Amazon actually using these tools to predict what kinds of products they should actually go make for all of us to buy. So it turns out that if you look across any of these technology companies, they all follow this pretty simple formula. And so the question we asked way back in 2013, when I was a grad student at the U, is whether or not we could evolve biotech into tech bio by following this same formula. But there's a bit of a challenge, and the challenge is in the data. We all know that if I pipette in an experiment and someone else pipettes in an experiment, we might get different results. I was not the person labeled as having golden hands when I was in the lab, but there were those in the lab who were labeled that way. And it turns out that biology is so extraordinarily complex, chemistry is so extraordinarily complex, that if you perturb the systems in slightly different ways, you can get radically different outputs. And so because our industry in some ways is still analog standard, because there is a little bit of a reproducibility crisis in the published literature due to us not actually tracking all of these metadata about the way we do our experiments, and because so much data in our industry is siloed, not only within companies, but between companies, we're in a position where it's relatively challenging to follow this technology approach in bio. And so we decided, and many other companies have decided, that the most important thing we could do to replicate this cycle was to actually generate our own data. And I'll throw in a little plug. We're going to host a mixer at our headquarters just a few blocks away on Wednesday evening. I'll have a QR code later if you want to sign up. And you can come check out all the robots that are doing these experiments. So we've got about 200,000 square feet where there are robots doing experiments, generating rich, high-dimensional omics data to the tune of millions of experiments every week. We're aggregating that data and then using our supercomputer to train machine learning algorithms, to train neural networks, to make predictions about relationships across biology and chemistry. And as I said before, the initial algorithms aren't very good. Biology is massively complex. But the most important piece of what we've built is that those predictions can allow us to then go test in our wet lab just a few weeks later what we've predicted from those algorithms. And that allows us to start creating this flywheel of learning and iteration. And so I'm going to talk a little bit more about it, and I'm going to start to get pretty nerdy. We'll go a little bit in the weeds. I hope you don't mind. And I'm going to start with a point solution. Because if you look at the field of tech bio startups, the hundreds of companies that are digitally native today, most of these companies have started with a point solution. They're working on a single problem in drug discovery. And that's how we started too. We helped pioneer and industrialize an approach called cell painting that allows you to take microscopy images of human cells and turn them through computer vision algorithms into a new kind of omics. And so if you perturb biology, you can measure the output of that perturbation, how it changes the system in lots of ways. And some of the more traditional omics approaches are things like transcriptomics, proteomics, metabolomics. But each of those captures just a sliver of what's happening in biology, and we wanted something that was more holistic, something that would give us a sense of the cellular state that aggregates all of that is happening in transcriptomics, proteomics, and metabolomics. And for us, that was phenomics. And what a phenomics essentially is taking lots of microscopy images like this. We stain cells, lots of different stains, looking at the nucleus, nucleoli, Golgi, ER, mitochondria, actin, and membrane. And then we use computer vision algorithms to turn every one of these images, tens of millions a week, into mathematical representations of biology. And it turns out that this is a super scalable, relatively inexpensive new kind of omics. And so this is a map of a CRISPR-Cas9 knockout of every gene in the human genome. So what I'm showing you here on each axis is every gene, and in red are phenotypes, morphological changes in the cells that are similar when you knock out that gene. And so you see that dark red line that goes down the diagonal, that's every gene compared to itself, and it's good that that's a dark red line. And you see some blue in various places as well. The blue means oppositeness in this high-dimensional phenotypic space. And if we zoom in on these clusters, I'll zoom in on a little tiny one, that little teal spot up there, you can start to rediscover known biology. So this is the JAK-STAT pathway. And what you see if you look at the bottom left is that we've knocked out JAK1, and you can see that the resulting phenotype, according to a computer vision algorithm, is most similar to knockouts of other related genes, like the receptors. And you also see SOX3 in blue, which is a negative regulator of that system. So we can use this phenomics approach to rediscover lots of known biology. And that's great because it gives us confidence that we're doing something that's working. And if we had more time, we could go through thousands of examples like this. But we can also use these tools to identify novel biology. We signed an incredible partnership in 2021 with Roche and Genentech, where we're mapping neuroscience for a decade together. And in the early days of that collaboration in 2022, the teams were using some of our maps to ask and answer questions about the integrator complex. And you can see that if you zoom in on another part of that map, there's a bunch of genes that are INTS and then a couple of numbers. These are all components of the integrator complex. But what was really interesting as we were doing that work is that there was a gene called chromosome 7 open reading frame 26, I believe, and it was clustering with all of these different integrator complex genes. And again, we've just used CRISPR to knock out all these genes. We organized the resulting phenotypes by similarity, and this relatively under-described gene was showing up as maybe part of this complex. And as the teams were doing this work, a fantastic publication came out that actually demonstrated quite elegantly that indeed chromosome 7 open reading frame 26 encoded a protein that was part of the integrator complex. And these kinds of things happen all the time at Recursion and all the time for people that are using phenomics and these other kinds of tools. But to do this well, to do this at scale, to map and navigate biology broadly, you really have to automate this. I talked earlier about the challenge of two different people pipetting and the different outcomes that you might get. For us, we believe that automation is one solution to these challenges. Not only because we can standardize everything, but also because we can measure everything. We know exactly how long every reagent was on every single well, and exactly what order we did each of the experiments. And when something goes wrong, we actually record all of the movements, and we can go back and look at the instant replay to try and figure out what happened. And so if you come on Wednesday night, you can check out this laboratory, and you can see what it takes to generate these kinds of data sets at scale. And as you do this work over the last decade, we've, I think, really helped pioneer the industrialization of this approach. And I want to talk about two pieces. One that gets a little bit into the machine learning weeds, but I think you'll like, and that is our foundation model in phenomics. So a foundation model, has anyone heard that term, a foundation model? Okay, so let's talk a little bit about ChatGPT. Who's heard that? Awesome, okay. So ChatGPT is a foundation model in language. They trained that model to predict what word should come next in a sentence when they've held out the rest of the sentence. That's how they train ChatGPT at scale. So GPT 3.5, GPT 4, all of these things were trained by just predicting what word should be next. But it turns out if you train a neural net to do that really, really well, you can actually start to do all kinds of emergent things. You can start to do math problems, or you can start to reason in some ways. And these are called emergent properties, and foundation models are the kinds of machine learning tools that help us train in a narrow way and then use the model in a broad way. And so we built a foundation model for phonomics. So rather than mask the word in a sentence, we did this by masking parts of the image. So you can see on the far right here, original images. We've got over 1 billion of these, so we had plenty of material to work with. You can see an original image, and on the far left there, you can see that we've masked 75% of the image, and we do this in a random way. And then if we, let me just go back. If we train a neural net to predict what the rest of the image should look like, we get something like what you see in the middle. So this is our most advanced foundation model at Recursion. Given just 25% of an image, you can see that it is able to reconstruct the rest of the image pretty well. They don't look identical, but those middle images look pretty close to the images on the right. Now, this is not a thing that we do a lot in drug discovery. It's not like we get our images and 75% is altered or something. But by training on this task, we were able to train this foundation model that is now able to help us with dozens of drug discovery tasks, predicting mechanism of action, predicting relationships across biology and chemistry. It turns out that just like with a large language model like ChatGPT, the same thing holds. And there's something else that we discovered when we were doing this work. It turns out that more data and more compute help you make better models. So you can see here the result if I train on 1/4 of our images with a small model, 86 million parameters. You can see that this image looks, there's like some splotches in there, and the cells are kind of not super detailed. And if I go to a bigger model, trained on 1/2 of our images and 300 million parameters, you can see that it gets better. And it turns out that this is called the bitter lesson in machine learning and AI. And that is that more data and more compute make your models better, all else held equal. And so far, this has sort of been true across every field in which people have deployed machine learning and AI tools. And that's both exciting and good to know that this is the kind of work we have to do, but it also means it's more expensive. We need more data and more compute in order to build better models. So what can we do with these models? I'm going to tell you a couple of stories about how we can take these models, identify relationships across biology that help us move medicines towards patients. And I'll start with a relatively complex assay. This is a PBMC-derived fibrocyte assay. So we've taken PBMCs, and we are deriving fibrocytes from them. And what we're trying to find is a small molecule that can mimic the effects of a very specific protein. And that protein essentially restores a non-fibrotic phenotype. So we're looking for small molecule mimics of this protein. And using this platform, we were able to test hundreds of thousands of molecules at many different doses and identify molecules that were changing this complex fibrotic phenotype measured in thousands of different parameters into something that looked much more like the healthy non-fibrotic phenotype. And so we were able to take lots of different molecules, identify some with different scaffolds that we were excited about, and you can actually do SAR using this phonomics approach. You can actually start to look at relative potency of all these different molecules. You can make changes to the molecules and retest them on the platform, and we were able to select a core that we were excited about. And then we were able to take this core and identify what we believe the target is. And unfortunately, we're on the way to the clinic with this particular program, and we're not yet sharing what the target is. But it's a novel target in biology related to fibrosis. And this was exciting to us because you can see target epsilon in the top right there. You can see 3 different molecules that have increasing potency as you go from bottom to top against this particular protein product. when we do a biochemical assay. But you can actually see that in the heat map, they go from orange to red to darker red because we're getting closer and closer to mimicking the effect of a CRISPR-Cas9 knockout of target epsilon with these molecules. And so we were able to hypothesize this was the target and then go test that biochemically. And this is a really fantastic use of phenomics. But you don't always have to look just for efficacy signals. You can also use all of the same data to simultaneously try to remove some of the unwanted features. So this confounding thing we noticed is that many of the molecules we were identifying that were having the right effect of rescuing this phenotype were also inhibiting microtubule formation. And we all know we don't want that, especially in fibrosis. So what we ended up doing was looking for molecules and cores that actually were, as you see in the green, not related to or not looking like knockouts of these different microtubule genes. You can see in blue that the compounds that biochemically had a greater than 30 micromolar IC50 against microtubule assays. Those showed up as not looking like microtubule gene knockouts. And so you can do SAR both for the good and the bad with this high dimensional approach, which we think is really, really exciting. And so we were able to use these tools to optimize the molecules through multiple rounds, develop a molecule that we could take into animal models, and then demonstrate to ourselves a really exciting outcome for this novel target in the context of fibrosis. But the reality is this one assay, this cell imaging assay called phenomics, is interesting in the context of all the work I just shared with you, but discovering and developing a medicine consists of hundreds of steps. And if you go off the rails on any of those, the program can die. And so we've moved from this point solution at Recursion to trying to build many different tools that all keep this philosophy of building these virtuous cycles of learning and iteration, but where we're generating different kinds of data sets, different kinds of algorithms, and then stringing these all together in automated workflows. So I want to share just a couple of those with you, and I'm going to start with our DMPK module. And if you come on Wednesday night, you can check this out. It's actually updated a bit since this picture. We're able to do 3 different assays today in both human and rodent cells at the scale of about 1,000 molecules a week at Recursion. And this means we don't have to send these out to a CRO to get the data. And so we're running all of our internal programs through this platform. But what's even more exciting is we're running hundreds of molecules every week through this platform to generate training data for machine learning algorithms that will help us predict the outcome of these DMPK assays based just on the structure of the molecule. And we believe this training set will ultimately be one of the largest in the industry, and will move from 3 of these assays to many, many more in the years to come. And I want to go back and share one of the assays we were working on a couple of years ago, and I think this one just is awesome because we've moved from individual primary human cells or rodent cells now into spheroids or organoids. And we were interested in the cardiovascular metabolic space and trying to ask and answer whether we could predict whether a molecule would be a hERG inhibitor through, or have a hERG liability through this assay. And so we took cardiomyocytes, we made these spheroids. You can see that they beat if you watch this video. We did it in 384 wells, and then we went to 1536 wells. So you can see on the right, I hope you can see it in the back, there's little tiny pixels that are blinking there. Those are 1536 well plates with beating cardiomyocyte spheroids in them. And we were able to use video to extract the signals from every one of those wells. And if you zoom in on those signals, you can actually start to measure all kinds of things about the electrophysiology of these spheroids. And what we were able to do was then cluster all of these tens of thousands of molecules that we ran through at many doses to ask whether or not they were perturbing the healthy beating cycle of these cardiomyocyte spheroids. And what we found was that the vast majority of molecules with a known hERG liability were actually moving the signal outside of its kind of normal cluster where you see these black boxes here. And so this is now training data that we can use to actually make predictions about whether a molecule might have a hERG liability. And of course, we can then go back in and test those predictions to ask and answer that question. Eventually, we hope, across billions and billions of molecules without having to do billions and billions of experiments. And you don't only have to look for liabilities. We actually worked on a rare genetic cardiomyopathy. You can see healthy beating cardiac spheroids on the top, diseased ones on the bottom. So we broke a gene important in this genetic cardiomyopathy disease, and we were able to identify molecules that restored a normal beating pattern in these cardiomyocytes. So I think it's really, really exciting to take this rich, high-dimensional data approach. But you don't have to stop at spheroids or organoids. You can go beyond that. And one of the things that I'm really excited about Recursion is our work that we call in vivomics. So we have a vivarium with thousands of cages, and in every one of those cages, there are multiple sensors, including cameras, in the lid. And these sensors are recording video of the mice or the rats inside the cage at all times. And what we're able to do with this is actually generate these live dashboards. So if you're a technician that works in our facility, you actually get a live alert if there's a problem with one of the animals. Maybe an animal is lethargic. You actually can decide, looking at the video, whether you need to go in and check on that animal. We can use fewer animals because we have such up-to-date monitoring. And we can do experiments in real time and actually extract data in real time using computer vision algorithms on the videos of these mice. And we are making decisions in near real time for our programs with these data. So this is an example of a program at Recursion where we had multiple molecules that we put into animals, and we were trying to select which of these we were going to move forward towards clinical trials. And so you see at the top, Rec 65029, we put into animals, and you see multiple day cycles there, and you see at multiple doses, including the efficacious dose. There's no change in this gross readout of animal movement, but there were also no important changes in the hundreds of other parameters that we can detect with this tool. Whereas another molecule like Rec 1170204 actually started to cause some worrisome changes in the mice according to our machine learning algorithms. However, we do not believe that our technicians would have noticed these changes if they were just doing the work in their normal course. We're detecting things that you might not uncover for 6 or 12 months until you go to do the toxicity studies and ultimately look at histology or something like that. And so we were able to deprioritize that molecule and prioritize another molecule, and hopefully saved ourselves 6 or 12 months on this program at Recursion. All right. But we don't want to stop at animals. We're not here today at Recursion to make drugs for animals. We're here to make drugs for people. And so we also want human data. And that's not something we've been able to generate at Recursion except through our own clinical trials, but we only have 5 of those right now, and so it's not scaled enough to get enough data. And so we actually recently announced a partnership with a company called Tempus, based in Chicago, who's been generating the world's largest DNA and RNA sequencing data set paired with clinical records from millions of oncology patients. So it's vastly bigger than something like TCGA. And we partnered with them to get access to all of that data, to bring it in-house, and to train ML and AI models not only on their data, but on our data at the same time. And this is truly harnessing the power of forward and reverse genetics to allow us to identify genes that are potentially encoding proteins that have some causal role in oncology that maybe are just too noisy or too subtle to pick out of either data set alone. So we've trained models that use both of these data sets, the cellular data we generate and the patient data from Tempus, to identify potential causal drivers in oncology. And we only signed this about 4 months ago, and about 8 to 10 weeks into the partnership, the team had already used these data to predict 2 relatively novel drivers in non-small cell lung cancer that you just weren't able to get from either data set alone. It was only through the combination of these data where you could start to identify these signals. And we're now working to evaluate whether gene A and gene B might actually be novel drivers of non-small cell lung cancer. Really, really exciting work. But not everything we do at Recursion has to come with its own data set. We also do use public data sets. So this is an example of a tool we recently announced called Mole. We've actually published this. You can go use it. And we trained these neural nets using graphs where we basically give a tiny piece of the structure, so one atom and the surrounding atoms, for 842 million structures into this self-supervised system. And then we fine-tune the model on data across 500,000 structures where we have activity in about 1,300 different ADME tox assays. And what we're able to do is train a generalized model, a foundation model, that's not good at predicting just one tox readout or one ADME readout. It's actually state-of-the-art in the Therapeutics Data Commons benchmarks against 10 out of the 22 measures. And this is pretty exciting because if you go look at Therapeutics Data Commons, what you'll see is that people have trained really cool ML and AI models, but in general, they've trained those models on each of the tasks individually. So you've got 22 different best models. And we've been able to train one model that actually makes some of the best predictions across many of these different readouts in ADME tox. And I think this is where the field is going. We're building foundation models, generalizable models that will help us make broad predictions about lots of the ADME and tox elements that we want to understand as we're developing a molecule. All right, I'm going to share one last example. This is one of my favorites. And then I'm going to talk a little bit about the future. So this last example is one that's near and dear to my heart because I've been asking the team for years, why can't we just do a whole genome, every gene by every gene, CRISPR-Cas9 knockout experiment to try and understand the gene networks that underlie biology. And it turns out the reason is that if you do every gene by every gene with a single replicate, you're looking at 199 million wells. So in our last 10 years at Recursion, we've done about 250 million experiments on our phenomics platform. So this is technically feasible work, but you're going to spend $100 or $200 million to generate this entire data set. But imagine how much we could learn. Just look at the world of yeast biology and what we've learned from synthetic lethality screens in yeast. Imagine if you could do the equivalent of that for every human gene against every human gene. And you could not just read out whether the cells lived or died, you could actually read out hundreds or thousands of dimensions of changes in biology with this omics approach. I think you could start to really untangle the web of biology. And what if we did 3 genes? So every gene by every gene by every gene. Now you're talking about 1.3 trillion experiments before you even do replicates. That's getting into the realm of crazy. And if you start thinking about time course and multiple genes in the network and all the different chemicals, you start to kind of feel like this guy, like the interactome of biology and chemistry is just so massive that there's no way we're going to brute force explore every possible combination. And so we need to take a different approach. And we believe that the future is active learning driving decisions that are happening in the laboratory. So what this means is that rather than each of us deciding what experiment we should do next, we should be using ML and AI algorithms to ask what would be the experiment that we do next that has the highest probability of gathering important information. And to test this idea, we did a small experiment. We did 50 genes by 50 genes with double knockouts. And we asked how well did we predict what actually happened when you did a 2-gene knockout from our prior 1 gene by 1 gene data. And in blue, you see good predictions, and in yellow, you see bad predictions. So we physically did the experiments, and you see, hey, our algorithms were pretty good. We predicted what would happen when you knocked out 2 genes together just from the single gene data, except in a few spots. And so then we trained an algorithm to take a little bit of data and then decide based on that data what experiment to do next. And so on the bottom, you see random experiment choice, like which gene you knock out with which other gene. And on the top, you see an active learning approach, where through each cycle of experimentation, we're making a decision about what is the best experiment to do next. And you see the much steeper blue line, the dark blue line. That is how much information out of all the possible information you can get that you are gathering with this active learning approach. And I think this is really important for all of us as we think about experimentation at scale, understanding the relationships across this huge interactome of biology and chemistry. We're going to have to start using ML and AI tools to not only analyze experiments, but to design and run experiments. And that's going to be a paradigm shift. And I think that takes us to the future of tech bio. The challenge today at a company like Recursion, or a large pharma company, or any of the hundreds of tech bio companies that are growing, is that we've moved beyond point solutions, and we're now building many different tools. And these tools are improving week after week, month after month, year after year. And it's actually hard to keep up, even if you're a computer scientist, with the latest version of every tool. And if you've got dozens of tools like we have, it's really hard to keep up with which is the latest version of the tools, which one you should use. And unless you're a data scientist or something like that, it can be even hard to know how to use each of these tools. So if we're looking for a new chemical entity, for a known target, maybe we use this combination. Or if we're looking for a drug for a novel target, we use a different combination. But ultimately, it's getting too challenging to stay up to speed. And this is where we believe large language models, like ChatGPT, but different, are going to make a fundamental difference in the biopharma industry. Rather than using large language models to go search all the world's literature and then come hallucinate about some results to all of us, what if we could use them to interact with us to help orchestrate the use of all of these tools? And so a couple of months ago at the JP Morgan Healthcare Conference, we hosted an event with Jensen Huang, the CEO of NVIDIA, and we unveiled a tool we call LOW, which is a large language model orchestration work engine. And this is a tool that allows any of our scientists or any of the scientists at our partners, or even people on BD or finance teams or working in supply chain, to interact with our entire operating system and all of our tools just through natural language. So in this web app, you can type in something like, provide a list of 5 targets in lung cancer, or give me molecules that are predicted to bind to RAF1, or sort the molecules in this list from a prior experiment by solubility, or order compounds from some algorithm using the API from one of our suppliers, or design an experiment and run an experiment. And what it means is that we're moving away from having to be an expert in data science or in biology or chemistry, and you just have to be an expert in understanding the process, the patient, the goal, and use all these incredible tools in the background. And what we believe, and I think many others in the field, there are a couple of large pharma companies actively developing tools a little bit like this, and 3 or 4 other small companies developing tools a little bit like this. What we all believe is that the future is going to look pretty different. And what we sense is coming is a revolution that is not unlike the revolution of the late 70s or early 80s in personal computing. So I'll probably date myself a bit here. I was not alive when the Apple I came out, on the left, but maybe some of you were, and maybe you actually played with this tool. But if you were not a computer scientist or like an electrical engineer, this was pretty intimidating. You had to understand command line, you had to be able to be comfortable with a circuit board and putting this whole thing together. This was a real challenging tool. And as such, the impact was relatively limited. And then a few years later, in the early 80s, the graphical user interface came out. The Apple Lisa came out. And all of a sudden, the power of a personal computer was available to orders of magnitude more people than it had been when it required that expert understanding. And we believe that just like the advent of the graphical user interface, what is happening right now over the next 2 or 3 years is the advent of discovery user interfaces, not only at Recursion, but at a handful of other companies around the world. And that this will revolutionize our industry much in the same way that the move to GUIs revolutionized the personal computing industry. And so I would advise everybody not to go learn to code. You don't need to anymore. You're going to be able to use natural language, your own thought, to be able to interact with tools at your company or our company or any of these companies, and you're going to super magnify the amount of science that you can do. You're going to eliminate toil from your work. And we're going to be able to advance more medicines to patients more quickly thanks to tools like this. So if you want to learn more about what we're building, I promised that I would flash this QR code. If you can't get a picture of it, you feel free to go to our social media at Recursion, and you can see where we're hosting the event just a few blocks away at our headquarters. We would love to host not all of you, because you can't fit. Our building is not as big as this, but as many of you as can join on Wednesday evening. Would love to host you. You can take a look at the robots, talk to our team, and learn more about what we see as this revolution that is coming to our space. And with that, and about 10 minutes left, I will pause. Thank you all for your attention. And I think we have a couple of mics that are out, so feel free to come up and ask a question. All right, is everybody going to be bashful? Perfect, yes.
Wonderful talk. Thank you so much. I'm wondering, in addition to the transcriptomics as well as the 2G knockout models, have you also considered factoring the epigenetics into the training of the models? Because as you know, some of these interactomes is tissue and age specific. And also, have you thought about factoring microbiome, which is also a complexity of human biology, into your future strategic planning?
Yes, great question.
Thank you.
So the epigenetics is absolutely something we and many others are working on. We're not working on microbiome at Recursion, but there's many other companies that are. And I think the challenge, of course, will be how do you aggregate different layers of data and make those data relatable, not only across a data set as it grows over time, but between those different data sets. And it's almost like the Google Maps problem, where you had aerial maps, you had data from cars driving, you had traffic data, and they had to integrate all of that into a cohesive solution. And that's the thing that a lot of us are going to face in this field. Thank you. Great question.
Hi, Chris. Very inspiring talk. Thank you. My question is, and I loved your idea of using human cell phenomics coupled with digital vivarium to collect data that would predict toxicity 6 months later. Are you able to convince regulators using a small molecule and this platform approach that we don't need to do, or we need to do very reduced animal toxicology studies like a checkbox exercise? And how is that uptake on the regulator side?
That's a great question. So I think it is far too early for us to propose not doing those studies. I think what we will find is that the goal of the tools we and many others are building are to help make better decisions earlier, so we move failure earlier in the process. So you've all seen the biopharma drug discovery funnel, right? It kind of looks like a V. And of course, the cost of drugs comes in the late stage failures, the phase 1, the phase 2, the phase 3 trials. When a drug fails there, you've now spent tens or hundreds of millions of dollars. And it's really damaging in terms of the economics of the industry and the ability to bring medicines to patients at a reasonable cost. What if you could simulate all of these things a decade from now or 2 decades from now, and then predict which molecule is going to work for patients and take it all the way to the market without attrition? It would look more like a T. And we think the optimal solution for the industry is to try and shape the drug discovery funnel like a T, to build algorithms that help you make these predictions up front. But we are not proposing that you don't still go do the experiments and, of course, take all of those data to the regulators, because if you're going to do an experiment in humans, there's an extraordinarily high ethical and moral bar that you have to meet. And so we believe, at least for the next decade or so, you'll still want to run the key experiments. You'll want to work with regulators to aggregate that data. And maybe, as we and others take hundreds of programs through the clinic, one day we will get to the point where we don't need to run those experiments. But I think that's multiple decades in the future. Maybe there are individual parts. Maybe there are certain areas of rodent tox that we may be able to eliminate earlier through some of these tools, or through things like organoid systems or tissue on a chip. But I don't think we're there yet, and we certainly aren't going to push the regulators to accept the output of some black box. I just don't think that will go well. Let's generate the data set to convince ourselves in the future that the technology is really there. I think we're going to go to, I think there's a line over here.
Okay, thanks. Really impressive. I'm just wondering, at the beginning when you showcased the Phenomics platform, you're using cells as model systems, and my question would be, what do you do to assure, or what kind of knowledge base do you build, maybe in the background, to assure that you've got in vivo relevance of the experiments that you got from those, let's say, model systems, which are cells and not actual humans, right?
That's a great question. So no model is perfect, but some are useful, I think is the term we like. One of the things we often do if we're working in a therapeutic area is we ask whether or not we can rediscover many of the known elements of biology using a model. So I'll give you a great example. A lot of our work, I came out of a vascular biology lab, and a lot of the work we've done at Recursion is in a workhorse cell type, human umbilical vein endothelial cells. And it turns out in areas like oncology, perturbing those cells does recapitulate what's known very, very well. It's about 80% of all of what's in oncology you can recapitulate in an endothelial cell. However, if you go into neuroscience, it totally falls apart. Like the predictions are awful. And so we announced a collaboration with Roche Genentech a couple of years ago to go build whole genome arrayed CRISPR knockouts and look at hundreds of thousands of molecules in neural iPSC cells. And it's early, we're only a couple years in, but we're seeing that those cells, not surprisingly, are much more predictive in the context of neuroscience. So again, I think it's just asking and answering these benchmarking questions, and then of course, moving to more complex systems. You just use it almost like a coarse filter. See if you can find something interesting from the system, then go to a more complex system, and keep doing that approach until ultimately you have a medicine that you're excited about. Thank you. Let's go to this one.
Hi, thank you for that talk. I'm Beck Petroff, and I'm actually a current Science and Technology Policy Fellow. So one of the things I was thinking about throughout, especially the end, is how my favorite part of being a scientist is being able to be creative and thinking and planning about what's next. And I didn't realize that machine learning and AI was coming for my job too. So I'm wondering what you have in your mind as far as how we can prepare the next generation of scientists to still be literate in science and be able to use these tools as well.
Yeah, it's a great question. And I think there's two extremes in outcomes here. One outcome is that, and I'll make these like extra scary sounding. One outcome is that ML and AI totally takes everybody's jobs across all industries for the next decade or 2 decades, and like none of us know what to do. We all become, I don't know, something that's not what we're doing now, okay? I don't think that's gonna happen. I think what's more likely, and what history has taught us about all of these tools, all tools that have changed humanity, that have led to industrial revolutions like the one we're going through now, is that these tools magnify our ability to do our work. And what I believe is likely is that the things that will be most important for humans is critical reasoning, creativity, caring and compassion. Maybe for physicians, we're not gonna have to memorize everything you need in step 1 and step 2 and all of that because AI will help us. But maybe bedside manner will become the differentiator for a great physician again. It'll be about empathy and caring and compassion. And so I think we will adapt. And what I would suggest to people is don't worry about learning to code. That's the first thing. Human language is gonna be what we need. But get like a classic education. Read the great works. Try and just learn how people think and how you can do critical thinking, because ultimately these tools are going to empower people who are creative, who can do that kind of critical thinking to do even more than they could have before. And so it'll be the people who have those tools, who have these critical reasoning skills, who can leverage tools like this to do even more for patients, even more for society. So I think it's in a way back to the early days, back to like a classic education, and maybe not so much into like the deep STEM fields. Of course, you'll have to learn a lot about biology and chemistry to be in our field, but you won't have to memorize all the little things. You won't have to do so much of that kind of deep toil. So that would be my advice. I hope that's helpful.
Yeah, thank you.
Thank you. Back over to this one.
Yes, hello. Thank you very much for the talk. Very nice. The question I initially had was taken because I think that's the most important about actually the cellular models.
The cellular models, yeah, that's a great question.
Exactly,
Yeah.
that's very important.
But you've got to back up.
I have several strings, but I will not. When you were showing your numbers about how many experiments you would need to map a relationship with your, and showing this trillion figure, I think you have to consider a trillion per cellular model, because a relationship in one model might not exist in the other, just because relationship in the different cell types, depending how they're organized within the body, might not exist in all. So that's the first thing. And the second important question, very quickly, do you think the supply chains within the biotech field is currently established to actually support this
No.
kind of effort?
No, I don't. So your first question, I 100% agree. That's why that guy's head exploded.
Yeah.
Because the interactome just gets too vast if you start thinking about all the different perturbations. So you'll need these tools. Supply chain, this is super important. Biology is so complex. We struggled for 6 months in the early days because our cells started changing in a way we didn't understand. And it turned out that a supplier to our supplier of tissue culture media had changed the source of the salt in one of the buffers. And that changed all kinds of biology. And so we now, it's not as glamorous as some of the other things I showed you, but we've probably got the most robust supply chain team in the world for a company of our size, because we have to be extraordinarily careful that we are tracking every single change in the reagents and measuring all of those things as well. And reagents change over time, but you have to be tracking those things, and I think that's probably one of the big drivers of the reproducibility crisis that we see in published literature, is just supply chain. And I think with that, we have 6 seconds left. So thank you all so much. I hope you enjoy the rest of the event. And thanks for coming to Salt Lake City.
I guess I'm not really. So thank you all for coming to the plenary. And we have a session in this room at 9:15, so we encourage you to move on to your next session so that we can get set up for the next set of talks. Thank you.